Okay, good afternoon. It's 2 p.m. Right now, and my name is Elisabeth Binder, and I would like to welcome you to this last but not least plenary session of this really exciting conference. The lecture will be given by Professor Katharina Domschke. Katharina is chair of psychiatry at the University of Freiburg. She has studied both medicine as well as psychology at the universities of Münster, Trinity College, Dublin, the Boston University, as well as Maastricht University. And from all her education, she has really generated a tremendous research career and received many awards for her research, including membership at the German National Academy of Sciences. Her research really focuses on understanding genetics and epigenetics of mood and anxiety disorders. I think what is really unique about her research is that she always has a translational angle going forward to clinical application. And I hope that she's going to present some of her exciting work in that direction at this lecture now. So join me in welcoming Professor Katharina Domschke.
Professor Binder, dear Elisabeth, thank you very much for the kind introduction. Ladies and gentlemen, dear colleagues, it's my great honor and pleasure to conclude this most exciting and inspiring conference this year in Milano with yet another exciting topic, and that is epigenetics. Epigenetics at the interface between stress and neurobiology, between the environment on the one hand and the genetic level on the other hand, between nurture and the famous nature. And as you can already feel and see, epigenetics is thus of great relevance with regard to psychiatric, with regard to mental disorders, and today with regard to anxiety and affective disorders. As you all know, as clinicians, as epidemiologists, as basic scientists, anxiety and affective disorders are the most common, the most frequent mental disorders among all. And these are the famous Wittchen data from the European Neuropsychopharmacology Journal, showing that unipolar depression and anxiety disorders affect up to 100 million people in the EU within 12 months. And they are not only very frequent disorders, they are also highly ranked regarding chronicity. So they take the highest rank or the 6th rank among all disorders, including somatic disorders, regarding the years lived with disability, the YLDs. And as you can imagine, therefore, they confer a great individual burden as well as a societal burden, a socioeconomic burden, basically, with very high direct, indirect, and cumulated health costs. There are evidence-based and effective treatments available. We have accumulated evidence here in the World Federation of Societies of Biological Psychiatry guidelines for the treatment of anxiety disorders quite recently, and you can treatments available. We have accumulated evidence here in the World Federation of Societies of Biological Psychiatry guidelines for the treatment of anxiety disorders quite recently, and you can also read about the world guidelines on the treatment of depression in another article in this journal. So mostly SSRIs and SNRIs are the first-line pharmacological treatment options in both disorders, which already alludes to a shared common biological trunk in the pathogenesis of those disorders. And on the psychotherapeutic side, the cognitive behavioral therapy is the way to go, the first-line way to go in those nosological entities. However, the patients and we as clinicians are not rarely faced with the challenge of treatment resistance. As you can see here from those Dutch data, within the time course of 9 years, about 30% of the patients do not respond sufficiently to those first-line treatments, with many of them remaining intermittently recurrent or even consistently chronic. And the same applies to the phenotype of depression, also here about 30% of the patients do not respond to initial treatment. So we are faced with the clinical challenge of treatment resistance in anxiety and depression. And this is why we very recently assembled a consortium of international experts, and we performed a so-called Delphi consensus method guideline, where we digged deeper into the problem of treatment resistance, in this case of anxiety disorders, to define what treatment resistance actually is and how to face this challenge. What ways are there to go to tackle these problems for those patients for which we have to try harder, both in a basic science way and a clinical way. And what 100% of the experts could agree with was that the development, the research into biomarkers would be a promising way to go. Why biomarkers? Well, if we're looking at anxiety disorders or affective disorders, we're looking at a, let's say, smoothie, where several ingredients are mixed together and you cannot really identify or recognize the ingredients that have been put into the mixer. So it would be much more favorable to identify for each and every individual patient or proband or human being the constellation of risk factors before the disorder manifests itself, so we can offer preventive measures. And also with regard to treatment, we're treating the smoothie with a one-size-fits-all program. So we have a limited array of treatment options, and every patient gets approximately the same treatment. It would be much more suitable and probably much more efficacious to offer individualized, tailored, precision medicine treatment to the patients according to their individual risk factor constellation, according to their biotype. And this is where biomarkers come in. First of all, biomarkers can help in identifying a disease risk. So which person is at an increased risk for developing anxiety or affective disorders, so that those probands, those human beings, can be offered selective or indicated preventive measures. Second, biomarkers can help to predict treatment response before a particular medication, a compound, or a psychotherapy is offered, so that patients do not have to go all the way through a frustrating treatment trial and then try the next medication or a combination of medications, have side effects. So it would be very helpful if we had indicators of which patient responds to which treatment best. And third, biomarkers can also be used as biomechanisms. So we can investigate the biomechanisms underlying treatment response or non-response, which can then inform the development of innovative, novel compounds or psychotherapeutic applications, or the combination of both. And this is what I'm going to talk about today. Now the question is, which biomarkers are we looking at? And this is not a new story. We've been looking at biomarkers for decades now. And we and many, many other groups around the world have spent millions or even billions of dollars investigating biomarkers on several levels, on the genetic level, on the neuroendocrine level, on the neuroimaging level, also in interaction with environmental factors, which in mental disorders obviously interact in constituting the overall disease risk in a complex genetic manner. However, if we look at the success of this endeavor, the sobering truth is, and that is what we found in a very thorough review that we've just published last year, that this search was not very successful. So looking on those levels I've just described did not result in viable biomarkers, as we summarized here. An enormous societal investment over the past 50 years has identified numerous candidate biomarkers. However, to date, the overwhelming majority of these measures have not been proven sufficiently reliable, valid, and useful to be adopted clinically. So this is where we stand. So in the search for viable, applicable biomarkers useful for the clinical application, we might have to look at another level. And one of those levels could be the level of epigenetics. The level of epigenetics is located in between the genetic level, as I said in the beginning, and the environmental level. So it is a mechanism, a procedure, a biochemical way where the environment speaks to the genetic level. It is at the intersection between, as I said, the genes and the environment. Epigenetic mechanisms are crucially involved in governing gene function. So they govern to which degree a particular gene is transcribed and then translated into the protein of interest. Epigenetic mechanisms are not only housekeeping mechanisms, but also, to a certain degree, dynamic. They are flexible, they can change, they can adapt. And if they can adapt to environmental challenges, they can also maladapt towards a risk status. So that makes them very interesting markers in the context of diseases, such as our diseases, that are located at the interface between biology and environment. And this brings me to the last and very interesting point. Epigenetic mechanisms have been shown to be responsive to the environment. In many animal studies, it has been shown that stressors, for instance, can crucially alter epigenetic patterns. And this is actually the mechanism that seems very interesting in the context of complex genetic disorders, such as mental disorders. So this is actually indicated by this bidirectional arrow here. So there is a back and forth between epigenetics and the environment. So now I cannot spare you a little bit of biochemistry. So what are epigenetic mechanisms? I'm only introducing two epigenetic mechanisms to you today for the sake of clarity. First of all, you know we have chromosomes, and the chromosomes are composed of the chromatin, where our genes are arranged, the DNA strand. And the DNA strand is very long, so it has to be wrapped around certain protein structures in the cell. And those protein structures are histones. Those are octamers of proteins where, just imagine a ball of wool, the chromatin thread is wrapped around, and you can imagine that knitting this thread of wool is very easy if this thread is not very tightly wound, but loose. And it's very hard to access this wall, this thread of wool, if it's tightly wound in this ball of wool. And the degree to which the chromatin is wound around those proteins is determined by histone modifications, such as acetylation, methylation, or phosphorylation, for instance. I won't be talking much about this process, but mainly about another epigenetic mechanism, and that is the direct methylation of cytosine bases of the DNA itself. And this occurs on cytosine bases, mostly when they are located adjacently to guanine bases. So if there is a dinucleotide of cytosine and guanine, which you often call CpG. So this methylation also crucially impacts the degree of transcription of the gene. And it does so in a differential way. And you just have to understand this principle, and then enough with the propedeutics, but this you have to understand in order to follow me through the next slides. So, very coarsely spoken, if the promoter region, so the governing region in front of a gene body, is methylated, as well as the first exon and maybe also parts of the first intron are methylated, then the gene is sort of silenced. It is turned down, it is dimmed down in its activity, while if the exons that are following the first exon are methylated, the inverse is true. So if they are methylated, the gene is active. So, enough of biochemistry, let's dive into what I promised, some examples of how do epigenetic factors contribute to maybe being disease biomarkers in affective and anxiety disorders. I am just presenting 2 examples of candidate genes to you, just for you to understand the principle for an illustrative reason. Of course, there are many more genes that have been investigated by us and worldwide, but just for you to understand the idea. First of all, the monoamine oxidase A gene, the short MAOA, and I think 70% of you are clinicians and are very familiar with MAOA inhibitors, which you give sometimes for the treatment of depression and sometimes also in case of social phobia patients. Why? Because MAOA is crucially involved in the metabolism of catecholamines, of serotonin, of norepinephrine. And those, of course, are the prime suspects in the pathogenesis of those disorders. So MAOA metabolizes those serotonin and norepinephrine molecules. So the more active the MAOA is, the less catecholamines are available in the synaptic cleft and vice versa. Prime suspect in our diseases, as I said. The gene coding for the MAOA is structured as every gene with a promoter region and exons and introns, and there is a CPG island, meaning that this is a region where there is a multitude of CPGs, so cytosine bases close to guanine bases, that can be methylated or not, encompassing the exon 1 and parts of the promoter region. So, according to a hypothesis, methylation of this CPG island should lead to a silencing of gene activity. In order to prove this hypothesis in this particular case, we performed a luciferase assay where we cloned the promoter of the MAOA gene in front of a gene which can be measured for its activity very easily, the luciferase in a luciferase assay. And as you can see here, if the promoter region and first exon region was methylated, the gene was actually really silenced, while in the case of non-methylation, as shown here, the gene was quite normally active. So, hypothesis corroborated, and we can now go to the clinical samples. So we investigated a sample of patients with panic disorder, short PD in black here, as compared to a very carefully, and this is very important in the context of epigenetics, because there are many confounders, carefully matched sample of healthy controls. And as you can see here, the MAOA promoter was significantly less methylated in patients with panic disorder as compared to healthy controls. What does that mean biochemically? Let's go back to our synapse. If you have, in case of the controls, a highly methylated MAOA promoter, according to hypothesis and according to the luciferase assay, the gene is then relatively silenced. So less transcribed and the protein is less active, so MAOA metabolizes less catecholamines, so there are more catecholamines available in the synaptic cleft. The opposite is true with patients with panic disorder. Here you have a decreased methylation, meaning that the gene is more active, MAOA catabolizes more catecholamines, and you end up with a situation of a catecholamine depletion in the synaptic cleft. And again, you clinicians will recognize this term. Catecholamine depletion is one of the hypotheses underlying our idea of the pathogenesis of anxiety and depressive disorders. So, biochemically quite plausible in panic disorder as a potential pathomechanism, and accordingly, colleagues from Sweden also found the same pattern in a sample of patients with depression, here in dark gray, as compared to healthy controls in light gray. Also, in those patients, they found a decreased methylation of the MAOA promoter, probably resulting in the same biochemical mechanism, pathomechanism in this case. So, we can state at this stage that epigenetic mechanisms seem to be probably pathogenetically relevant for anxiety and affective disorders. The more interesting question is, how does this mechanism relate to the environment, as I've introduced to you before? So the next question is, do negative environmental influences maybe lead to an increased risk towards anxiety and depression via a demethylation and thus activation of risk genes. So we looked at the sample of patients with panic disorder as well as healthy controls, and what we found was that there was an inverse correlation between MAOA promoter methylation and the experience of subjectively negative life events in the year before inclusion into the study. So if you have a look at the x-axis, go to the right, the more negative life events you have, the lower your MAOA methylation. So this might be one of the mechanisms how the environment, the negative environment, speaks to our genes, wakes up our risk genes, maybe via the modulation of epigenetic mechanisms. First example, check. Second example is the OXTR gene, the gene coding for the oxytocin receptor. And you all know that the oxytocin is our pro-social hormone. So it facilitates the interaction, the social interaction among human beings. And so we were interested in this gene, particularly regarding social anxiety disorder. So we looked at the gene, and this time we did not look at the region of the promoter and exon 1 of this gene, but the exon 3 region. So remember the biochemical propedeutics. Methylation in the middle of the gene leads to an activation of the gene, and a hypomethylation would lead to a downregulation of the transcription. And what we found was that patients with social anxiety disorders in black actually did show this hypomethylation of this oxytocin receptor region, particularly at 1 CpG, as compared to healthy controls, meaning that maybe the function of the oxytocin receptor is a little bit impaired, and thus oxytocin transmission, the pro-social function, is a little bit impaired in those patients, which is biochemically, again, plausible considering the phenotype of social anxiety. We followed up on this finding in the categorical phenotype of social anxiety, also in a subclinical phenotype, because in the end we're interested in identifying biomarkers that are not only evident in patients, but already in healthy probants, in healthy people, that can be identified at persons at risk for preventive interventions. So we looked at a subclinical sample of persons that were characterized for the social phobia score on the social phobia scale, which is a subclinical symptom scale. And what we saw that, again, persons with a subclinically elevated score for social anxiety also showed a relatively decreased methylation of the oxytocin receptor. And we now took this finding to an intermediate phenotype level to follow it through the levels, so to say, and investigated whether this could maybe transported by cortisol interaction, because cortisol functions as a transcription factor in the core of the cell. So maybe the stress people experienced during a socially stressful situation can lead to an activation of the HPA axis, and via the induction of cortisol could then lead to a modification of those epigenetic patterns. And what we saw was that indeed during a trias social stress test, a social stressor, where you as the audience would frown at me at this point and not shake your head and leave the room because I'm telling nonsense. That is the trias social stress test. So a great social stressor. And indeed, under this situation, higher cortisol levels indicating higher social stress went along with a decreased OXTR methylation. And finally, we were looking at the amygdala as the central relay station, the fear center, so to say, in response to social phobia-related words, blushing, embarrassing, loser, audience. So probants were confronted with those words in the scanner, inducing social phobia symptoms. And also here we saw a correlation as we expected, that is to say a higher amygdala response, a higher neurobiological fear circuit response when there was lower OXTR methylation. So those were the two examples for how methylation patterns in candidate genes could maybe serve as biomarkers indicating a certain disease risk and could be used for the development of preventive measurements or interventions in those probes that carry those epigenetic risk patterns. However, you might have already identified that I was talking about candidate genes. So we are only looking at the candidates we were already coming up with. So there was a priori hypothesis that led us to the investigation of those candidates. However, most probably we're losing a lot of information if we're not applying a hypothesis-free approach, interrogating the entire genome in a hypothesis-free and therefore hypothesis-generating approach. And this is what we did in analogy to the GWAS that you're all familiar with. Remember the Manhattan plots that are shown everywhere? We did an EWAS. So instead of a genome-wide association study, we did an epigenome-wide association study, short EWAS, in panic disorder, and indeed identified, even though on a suggestive significance levels, only novel candidate genes, novel potential biomarkers indicating disease risk in panic disorder, and that is to say differential methylation, for instance, in the cilia and flagella-associated protein that is expressed in the brain. Now, if you have a close look at this slide, you can see that sample sizes are super small. Those are the sample sizes. Can I get my mic back? Can you still hear me? Yeah, you can hear me again. So even the mic was embarrassed about these sample sizes. Very small. However, those were the first pilot studies, right? There are only 4 of those EWAS around, and only very recently there is an EWAS published by John Hettema that looks at larger sample sizes. So we and John Hettema went on to look at larger sample sizes in order to increase the power of those investigations. And second, we went on to not only look at panic disorder, but to the entire group of anxiety disorders, applying a cross-disorder approach. So we teamed up with national experts in Germany at several centers and investigated a relatively large cohort of patients with anxiety disorders at 3 time points, before, after, and at a follow-up time point accompanying cognitive behavioral therapy intervention. And we teamed up with the Max Planck Institute of Psychiatry, with Professor Binder, regarding the recruitment of healthy controls. So we were able to, in a first research question, investigate in a case control approach the differential methylation between cases with panic disorder, with social anxiety disorder, and specific phobias versus healthy controls. And those were approximately 400 patients. Now you see there is a huge increase in sample size and about 300 healthy controls. And indeed, we now came up with a substantial number of significant hits that were significantly differentially methylated between patients and controls. And those are very hot data that are currently prepared for publication. So I cannot name you the hits, but I promise those are very interesting genes that are implied in this EWAS that you hopefully can read about very soon. So this is the first big part of the talk. How can epigenetic patterns be used as disease biomarkers indicating disease risk, and therefore indicative maybe for preventive interventions that are indicated or selective and can help reduce the manifestation and therefore the disease burden of anxiety and depression, anxiety disorders and depression. Methylated between patients and controls, and those are very hot data that are currently prepared for publication. So I cannot name you the hits, but I promise those are very interesting genes that are implied in this EWAS that you hopefully can read about very soon. So this is the first big part of the talk. How can epigenetic patterns be used as disease biomarkers indicating disease risk and therefore indicative maybe for preventive interventions that are indicated or selective and can help reduce the manifestation and therefore the disease burden of anxiety and depression, anxiety disorders and depression. Second research question, how can epigenetic patterns be used as treatment response biomarkers? How can they predict whether a particular patient responds well or not so well to a particular therapeutic intervention? Just one example on a candidate gene level. Again, as you're already familiar with this gene, the MAOA gene. We looked at a sample of patients with depression in this case. We followed them up for 6 weeks under a very strict treatment regime with SSRIs. We took the Hamilton Depression Score each and every week, and we determined the degree of methylation of the MAOA promoter in exon 1 before treatment started at T0. And what we saw was that when we performed a medium split in patients relatively highly methylated, relatively lowly methylated, we saw that methylation levels that were relatively high were associated with a very nice steady decline in Hamilton depression scores over the course of 6 weeks. So this is what we want to see. We want to see a treatment response decline in Hamilton depression score of about 50%. That is response. What we don't want to see is the red line, and those are the patients that were initially relatively highly methylated. And those patients, as you can see here, show a much less pronounced and impaired treatment response over the course of time, which was significant. Again, back to our synapses, does that make sense? Well, yes, those patients that were highly methylated have a lower MAOA activity, so they have a relatively highly amount of catecholamines available in the synaptic cleft. If you now give an SSRI, neuroscience applied, so this is the clinical relevance of this finding. If you now give an SSRI, you block the reuptake of those catecholamines, of the serotonin in this case, so you have very many catecholamines available for postsynaptic transmission. So this is the way we want to treat patients. However, if you have the constellation of relatively low MAOA methylation and thus high MAOA activity, and thus a relatively impaired availability of catecholamines in the synaptic cleft, it doesn't help a lot to give an SSRI, because if there is not a lot to block from reuptake, well, there's no mean in giving an SSRI. So maybe, and this is the clinical relevance, it would be helpful to give those patients that have relatively low MAOA methylation not an SSRI in the first place, but rather an MAOA inhibitor that in a personalized, individualized, tailored way targets the problem, the underlying pathogenetic, relatively highly methylated. And those patients, as you can see here, show a much less pronounced and impaired treatment response over the course of time, which was significant. Again, back to our synapses, does that make sense? Well, yes, those patients that were highly methylated have a lower MAOA activity, so they have a relatively highly amount of catecholamines available in the synaptic cleft. If you now give an SSRI, neuroscience applied, so this is the clinical relevance of this finding. If you now give an SSRI, you block the reuptake of those catecholamines, of the serotonin in this case, so you have very many catecholamines available for postsynaptic transmission. So this is the way we want to treat patients. However, if you have the constellation of relatively low MAOA methylation and thus high MAOA activity, and thus a relatively impaired availability of catecholamines in the synaptic cleft, it doesn't help a lot to give an SSRI, because if there is not a lot to block from reuptake, well, there's no mean in giving an SSRI. So maybe, and this is the clinical relevance, it would be helpful to give those patients that have relatively low MAOA methylation not an SSRI in the first place, but rather an MAOA inhibitor that in a personalized, individualized, tailored way targets the problem, the underlying pathogenetic mechanism, in this case of relatively low MAOA methylation. This is just a mind game. Please be reminded of that. This is not replicated yet. This is first findings. This is just the mind game of how epigenetics could help eventually in daily clinical practice to help our patients get a more tailored, individualized, personalized precision medicine treatment. So again, this was a candidate gene example. Let's move on to the genome-wide level, to the epigenome-wide level. And we've just finished a study in a larger sample of patients with depression who were also followed up for a treatment response to SSRIs and a larger array of antidepressants. And here we found on a genome-wide level 6 suggestive hits that hopefully are accepted very soon, which you can read about in the International Journal of Neuropsychopharmacology. Third point, how can epigenetic patterns, mechanisms serve as treatment mechanisms maybe, informing about the underlying mechanisms of treatment response or non-response. And again, we were looking at the MAOA promoter and exon 1 methylation because we repetitively found that patients with anxiety disorders were lower methylated at this MAOA region. Again, in this study we were performing here. But this study now took another turn because we offered those patients with panic disorder a 6-week cognitive behavioral therapy. And after those 6 weeks, we split the sample of patients with panic disorder into responders and non-responders. And about 50% of the patients responded, 50% not. And then we investigated MAOA promoter methylation before therapy and after therapy. And we're interested in whether the responders differed from the non-responders regarding the dynamics of MAOA methylation. And what we saw was quite fascinating because after treatment, responders, clinical responders to treatment, showed a normalization of MAOA promoter methylation. So they were not distinguishable from the healthy controls anymore. So they sort of recovered, not only clinically, but also regarding the epigenetic risk pattern. While non-responders did not increase in methylation, they stayed hypomethylated or decreased even a little bit more in methylation. What's the clinical relevance of this finding? Again, let's play the mind game. Well, we could maybe give those non-responders after 6 weeks of CBT an adjunct medication with MAOA inhibitors, which sort of helps, which catalyzes the treatment response by addressing the underlying pathomechanism of MAOA hypomethylation. Okay, again, candidate gene level, now epigenome-wide level. We looked at a sample of patients with panic disorder, now on an epigenome-wide level, and came up with novel candidates that might be involved in the moderation or even mediation of treatment response, for instance, the interleukin receptor 1 gene. And again, look at the sample sizes, very small pilot study. So we went back to our huge or larger treatment trial, coming up with over 200 patients that were longitudinally followed for DNA methylation dynamics. And here we saw 4 suggestive hits that changed in methylation along with treatment response. So in the end, one of the greatest minds in our field, Eric Kandel, might have been right in his controversial, back then, 20 years ago, controversial saying, insofar as psychotherapy is successful in bringing about substantive changes in behavior, it does so by producing alterations in gene expressions. So a reconciliation between psychotherapists, psychiatrists, basic scientists, and environmentalists on the other hand. And that is what epigenetics can do. It can reconciliate, hopefully, between the biological and the environmental level by showing that epigenetics is responsive to environment. It can predict treatment response. It can be modulated by treatment. It is malleable, and is maybe also malleable by positive life events, which I'll show in a second. But I don't want to be silent about the limitations of the field that we can discuss later on. Reproducibility, the comparability between the brain and the periphery, of course, the complexity of the epigenetic mechanisms that are not only methylation processes, but, as I said in the beginning, also histone modifications. So one of the next steps would be to look at histone modifications that, at least in animal models, have shown some promise as potential biomarkers and biomechanisms in stress-related, anxiety-related, and affective phenotypes. So they should be looked at in future human studies as well. And of course, as I said, if epigenetic mechanisms can confer a risk, they can also confer resilience. And what we've seen was that MAOA was not only hypomethylated in response to negative life events, but also showed a positive correlation if probants experienced positive life events in the year before inclusion into the study. So they are highly malleable. They can confer risk as well as resilience. Let's be one step ahead of the patients going the entire way from a risk status to the manifestation of the disease through bothersome treatment and then maybe to response, but in 30% of the cases non-response, and start with a preventive intervention where epigenetic markers could be indicative of a risk and could be malleable by a preventive intervention. Some treatment and then maybe to response, but in 30% of the field that we can discuss later on. Reproducibility, the comparability between the brain and the periphery, of course, the complexity of the epigenetic mechanisms that are not only methylation processes, but, as I said in the beginning, also histone modifications. So one of the next steps would be to look at histone modifications that, at least in animal models, have shown some promise as potential biomarkers and biomechanisms in stress-related, anxiety-related, and affective phenotypes. So they should be looked at in future human studies as well. And of course, as I said, if epigenetic mechanisms can confer a risk, they can also confer resilience. And what we've seen was that MAOA was not only hypomethylated in response to negative life events, but also showed a positive correlation if probants experienced positive life events in the year before inclusion into the study. So they are highly malleable. They can confer risk as well as resilience. Let's be one step ahead of the patients going the entire way from a risk status to the manifestation of the disease through bothersome treatment and then maybe to response, but in 30% of the cases non-response, and start with a preventive intervention where epigenetic markers could be indicative of a risk and could be malleable by a preventive intervention. So my last slide I have borrowed from Viktor Frankl, a famous Austrian colleague, which is well known to the psychotherapists among you. And I've borrowed this quotation because it fits well with the context if it's twisted a little bit. The original quotation is as follows. Between stimulus and response, there is a space. In that space is our power to choose our response. In that lies our growth and our freedom. And I twist this a little bit, took the liberty into between genes and behavior, there is a space. And in that space is our power to choose our epigenetics. And that lies our growth and freedom. So it is our power to a certain degree at least, to mold, to shape, to form our epigenetic patterns by preventive measures such as nutrition, such as stable social relationships, not smoking cannabis, as Yasmin Hurd said in the opening ceremony, as I'm saying in the closing plenary of this day. And with that, I would like to thank you for your interest and thank my beautiful, wonderful group in Freiburg, in Würzburg and Münster, my collaborators and the funding agencies, and I'm looking forward to your questions.
Yeah, thank you. Thank you very much for an excellent presentation. We actually have many questions, and we have some time, I understand, for you to answer them. So the top question actually is interested in the dynamics of methylation changes. You clearly showed that it is possible. Did you study any timelines of trajectories of changes answer this?
So the timeline we've looked at was 6 weeks, because this was the timeline of our cognitive behavioral therapy intervention. So it's relatively quick. We were not expecting that. But knowing from animal studies as well as human experimental studies, we can tell that epigenetics changes can occur as fast as 15 minutes. So if you do a stress test, the triasocial stress test, for instance, that I've introduced to you before, there are methylation changes within 15 to 90 minutes.
Okay. So that's good that the second question actually was that. The other one is, I think you showed some data related to that, but it's candidate versus the whole genome, and we know from genetics it's many genes contributing. How is that in epigenetics?
It's exactly the same. If you have looked closely at the genes, you've seen that the methylation differences are about 1 to 2 to 3% only between cases and controls. So this is only a very small effect, as it is with genes, right? We're looking not only at a polygenetic, but also at a polyepigenetic phenotype with mental disorders. So in the end, we have to go the exact same way as in genetics. That is to say, we have to come up with polyepigenetic risk scores probably, and they have to be correlated with genetics, probably, right? Because they interact to a certain degree.
Okay, thank you. There were also questions related to the effect sizes. I think you showed some very stable and replicable effect sizes, but they're very small. Can you comment on that? How that could relate to function or clinical relevance is the question actually here.
Yes. As I said, very small effects, right? 1 to 2% difference. On the other hand, you don't want to have a black and white, an on and off system here, because if you switch off MAOA, you might have cardiovascular problems or whatever. So those are very subtle changes that in looking at the entire array of epigenetic modulations together with genetic modulations and other neurobiological factors will then in the end constitute the entire risk factor panel. But leaving out epigenetics would leave out a substantial part, I think, right? So we have to add epigenetics to the smoothie, right?
Oh, okay. So another question was related to cell type specificity and how the changes that you find may be related to changes that could be seen in the brain.
Yes, that's the big controversy about periphery versus central processes. Well, let me talk first about blood and brain comparability. Of course, biomarkers have to be accessible. We cannot take out a piece of the brain and look for biomarkers there, obviously, not even in cases or controls or problems, and looking at longitudinal changes, even more impossible. So we have to rely on peripheral biomarkers, so we have to rely on blood or saliva. So we have to first show whether it's legitimate to use this biomaterial that is accessible for a window to the brain. And if I can get my slides back just for illustrative purposes, because this is a question that often arises, I'll start. So we need studies that have been done, for instance, by Nadine Provençal, comparing methylation patterns in the prefrontal cortex of rhesus monkeys versus T lymphocytes. She showed quite some correlation between those patterns. We can do PET studies in humans where we correlate the peripheral methylation patterns in the blood with brain protein activity as measured by positron emission tomography. And here also there was some correlation as we've expected for the MAOA gene, for instance. But this is not thoroughly done. With the Epigenome-wide analysis, we've done a statistical control for the composition of the blood. But of course, as John Hedema did in his last study, you can also look at the separated cell types. And there might be differences, yes.
Okay, well, I don't think the slides will come, but
too late.
Maybe one last question. Do you think it's warranted to look at drugs that interfere with epigenetic mechanisms as pharmaceutical approaches for psychiatric disease?
Yes, I would have loved to have my slides, but well, maybe you've visited the session on new medications in the auditorium, and they were talking about LSD1 inhibitors. This is an epigenetic drug, actually, that affects anger, aggressiveness, and bipolar disorder. And we also know that the drugs we're currently using, valproate, lithium, SSRIs, NRIs, actually alter epigenetic patterns. So they might actually be epigenetic drugs already, right?
Okay. Well, with this, thank you very much for a great presentation, great Q&A, and we will move over to the closing ceremony now.